Hello, my name is Ramiro Garza, and today I'll be presenting our research on finding the molecular links between abnormal myelin and impaired behavior in Costello syndrome. The RAS pathway is a pathway that allows for the communication between surface receptors and DNA in the nucleus of the cell to produce a cellular response, and the disruption of this pathway can cause neurodevelopmental disorders. Our research focuses on the mutation of the HRAS gene, which causes a disorder called Costello syndrome. Costello syndrome patients are predisposed to ADHD, autism spectrum disorder, impaired motor skills, and intellectual disabilities. Costello syndrome patients are also found to have myelin and white matter defects in their brains. Studies have shown that myelin is a major regulator for brain function and plays a role in learning. Also, it is proposed that issues with myelin can lead to neurodevelopmental conditions, particularly in Costello syndrome. As mature oligodendrocytes are the cells responsible for making and maintaining myelin, we hypothesized that mice carrying a mutation causing Costello syndrome in oligodendrocytes have impaired learning skills. Therefore, our objective was to define specific behavioral issues caused by a Costello syndrome mutation, specifically HRAS G12V, in oligodendrocytes. To study the impact of HRAS mutation on myelin biology and learning, we induced the HRAS mutation in oligodendrocytes by using a mouse genetic system in which injections with tamoxifen induces the mutation of HRAS in adult mice. To find learning issues caused by the HRAS mutation in oligodendrocytes, we subjected Costello syndrome mice to a test well known to be regulated by myelin function, the fine motor skill test called the complex wheel. From this test, we evaluated the maximum speed, total average speed without zeros, and motivation to run. All of these 5 parameters inform us of different aspects of the brain function of these mice. We used 2 protocols of HRAS mutation and analysis of behavior in the complex wheel. In the first protocol, we tested homozygous HRAS mutant mice. Before the mutation of HRAS with tamoxifen treatment, both mouse genotypes learned at the same rate in the regular wheel. At 9 weeks, we induced the HRAS mutation in mature oligodendrocytes by injecting tamoxifen. This was 1 week after starting the mice on a regular running wheel. At week 10, we introduced them to the complex running wheel, which has missing rungs. Tamoxifen treatment equally affected both genotypes and their running performance in the running wheel. Performance was also equally recovered after treatment. At 10 weeks, when the mice were introduced to complex wheels for 2 weeks after HRAS mutation, we observed that the maximum speed or motivation to run was not affected in HRAS mutant mice running in the complex wheels. Interestingly, the speed learning curve was significantly affected in HRAS mutant mice in the complex wheel. For our second protocol, mice were first injected with tamoxifen, causing the HRAS mutation. 10 weeks after the mutation of HRAS, the motivation to run on the complex wheel was unaffected in mutant mice as compared to their wild-type counterparts. However, the max speed and average speed learning curves during those 2 weeks were significantly different than the learning curves in wild-type mice. We removed the mice from the complex wheel, waited for 3 weeks, and reintroduced them to the complex wheel to test their memory. Upon HRAS mutation, the total average speed was affected only at 3 time points. However, the total learning curve was unchanged in mutant versus wild-type mice. Overall, in figure 2, the average speed and the max speed learning curves were significantly changed in HRAS mutant mice compared to their wild types. Our results show that mice with HRAS mutations have subnormal learning curves. The average speed and the max speed were lower for the mutant mice compared to the wild-type mice. Therefore, the importance of myelin in learning is highlighted. We propose that the HRAS mutation in oligodendrocytes contributes to the neuropathophysiology of Costello syndrome patients. Our ongoing work focuses on finding drug treatments to revert behavioral issues in Costello syndrome mice, which can positively impact the future treatments for Costello syndrome patients. Thank you so much.